This lecture outlines a new way of thinking about modeling and hypothesis testing, a class of functional mesoscale models for whole organisms, a scalable architecture for learning such models from data, and a roadmap for building the required technology and infrastructure. We will focus primarily on describing the proposed class of mesoscale models and the machine learning architecture employed for inferring such models. These are compositional models in which the components are artificial neural networks, characterized here as shallow, deep neural networks for reasons that will soon become apparent. The machine learning architecture is also an artificial neural network that serves to configure, train, test the mesoscale models, and that also serves as a simulation tool for generating hypotheses and performing experiments designed to test whether the model exhibits analogous biological properties. The idea of a neural network that learns another neural network is conceptually simple, but technically challenging to understand, especially for scientists not steeped in the jargon and technology of machine learning and artificial neural networks. And so the material covered in this presentation will include the necessary background to make the lecture self-contained. So what is a mesoscale model? A mesoscale model, in some contexts we substitute the term mesoscale theory, is poised somewhere between the micro and the macro scales. Such theories are intended to serve as a conceptual and explanatory bridge between the cellular and molecular theories on the one hand and the cognitive and behavioral theories on the other. Terry Sosnowski at the Salk Institute describes mesoscale theories that provide a compelling bridge between the extremes of scale evident in biological function as the holy grail of computational neuroscience. I like the physicist's use of the term emergent as it applies to the quantum field, kinetic particle theory, fluid dynamics hierarchy of abstractions, with different mathematics, terminology, and physical intuitions at each level. Emergent models make assumptions that simplify understanding and facilitate calculation. In the case of the mesoscale model discussed here, the primary simplifying assumptions and hypothesis really is that the microscale circuits employ ensembles of neurons because individual neurons are not particularly reliable computing devices. These individual neurons have to work together to achieve a composite level of reliability sufficient to ensure the survival of the organism. What's wrong with the three models shown here? They are perfectly fine expository models but do little to explain observable behavior, and their explicit appeal to computation is tenuous at best. In each of the three models shown in the inset, neurons are represented as distinctive icons, so that, for example, pyramidal neurons are shown in red triangles and interneurons as blue circles. Connections between neurons formed by axons, dendrites, and synapses are shown schematically, and the polarity or valence of those connections, whether excitatory or inhibitory, is similarly indicated by graphical convention. These diagrams illustrate intuitive explanatory stories, but fail to provide an explicit predictive computational theory, remain anchored at the level of cell types, and make little effort to connect with observable behavior at the macroscale. So what do I mean by a computational theory? The characterization of computation as information processing that was prevalent in the middle of the 20th century is still relevant today. Certainly neural computation involves the exchange of information, and the activity of neural activity is best described as a process. However, I prefer the definition suggested by Peter Denning that employs the more specialized concept of a representation as an alternative to the colloquially overused and technically ambiguous term information, and defines a computational process in terms of transforming such representations. Neural circuits that represent various aspects of physical, sensory, and conceptual space abound in organisms throughout the animal kingdom, and, now it appears, in plants and other sessile organisms as well. Insects, mammals, and most eukaryotes are adept at utilizing, updating, extending, and combining representations to assist in everything from simple navigation to cross-referencing sensory impressions to create rich multimodal encodings of experience that facilitate mating, hunting, foraging for nutritious foods, and avoiding toxins. We'll start by describing what an experimentalist or model builder must specify in order to infer a mesoscale model from data, and what he or she can expect as output. A mesoscale model requires as input a source of structural data, which in our case corresponds to an annotated connectome. By connectome, I mean the equivalent of a 3D reconstruction of all the neuropil within the target tissue, which could be a complete brain of an organism or subcircuit of an optic lobe of such as an optic lobe or of the fly. Practically speaking, we are interested in the fly brain, 100,000 neurons and approximately 10 million synapses, and the similarly sized brain of the larval zebrafish. Regarding the connectome, we are interested in the graph consisting of neurons as vertices and edges that represent a suitable summary of the connections between pairs of neurons. In our case, we are interested in what is formally called a multigraph, in which there is a directed edge from A to B for each synapse, such that A is the presynaptic neuron and B is the postsynaptic neuron. It's important to note that the adjacency matrix by itself is not sufficient for our purposes. The connectome graph is embedded in a 3D volume whose geometry approximates the spatial properties of the original tissue. In particular, we require a spatial database that includes the coordinates of every neuron and every synapse within the tissue. In addition, we require cell types, morphology, and synapse metadata for calculating estimates of connection strength, such as the vesicle counts and synaptic cleft measurements. To infer functional models, we require a source of functional data. In this case, we assume state-of-the-art methods for recording from all of the neurons in the target tissue, such as the use of genetically modified organisms that express fluorescent proteins indicative of functionally relevant activity, and two-photon excitation microscopy to record estimates of local calcium flux as a proxy for electrical activity. The result is a time series of point clouds, where each point cloud is embedded in a 3D volume the same size as the connectome graph embedding. Within a point cloud embedding volume, each point source represented in the cloud corresponds to the fluorescent emissions emanating from a single neuron cell body, generally confined to the cell nucleus or the area adjacent to the axon hillox. Since we are attempting to bridge the gap between the microscale cellular and the macroscale behavioral scales, we need a source of behavioral data. For now, suffice it to say that we expect to acquire high-speed video of the target as part of the experimental protocol. Both functional and behavioral data will contain timestamps to facilitate alignment. The raw footage is bulky and inadequate as ground truth without additional annotation. And so researchers working on organisms like C. elegans, Drosophila, and zebrafish analyze high-speed video to identify stereotypical microbehaviors referred to as behavioral modes. For example, a simple reaching behavior may have preparation, initiation, extension, and termination modes, and then decompose complex behaviors into their component modes so as to more precisely align neural state changes with behavior. Finally, we assume that we are given an inductive bias that constrains the class of models and facilitates generalization. There are only two types of inductive bias for which there is theoretical justification for their ability to generalize from observed instances of a given class to instances not appearing in the training data. They are minimizing models, which corresponds to Occam's razor, and maximizing margins, an example of which is a support vector machine. In our case, we use the method of minimizing models to focus attention on a restricted class of configurable neural networks that are used to instantiate what we refer to as functional modules. In the literature, the term functional module typically refers to relatively small circuits purported to exhibit modular function. We extend the term to apply individually to each subcircuit in a disjoint set of subcircuits that together comprise a single connected circuit representing the entire target tissue. For our purposes, a functional module consists of a domain, a configurable network, and an interface that serves much the same purpose as an application program interface or API in software engineering. A mesoscale model defines as output a partition of the recorded point sources into possibly overlapping functional domains, where we use the word domain as it is generally used in mathematics to represent the domain of a function, but then abuse the notation somewhat when we blur the distinction between the domain and the range of a function. We justify this simplification since the functions we are discussing here are intended to predict the state vector of some subset of all neurons at time t from the sequence of such state vectors from time t0 to time t minus 1. And so it is technically more precise to say that the domain is the set of all contiguous sequences of state vectors starting at t sub 0, and the range is the set of all state vectors. In addition to defining the domains for each functional module, we also have to map each functional module to a configurable neural network. Here we separate the identification of functional domains from the assignment of configurable networks, but it should be apparent that all three components of a functional module, including its domain, network, and interface, are inextricably dependent on one another and therefore must be inferred jointly. Finally, a mesoscale model defines a sparse functional basis that serves to explain all the computations performed in each domain. Since this concept might be unfamiliar to some, I'll need to unpack the concept with some care. It is one of the key features of the proposed class of mesoscale models, providing as it does our ability to identify the distribution of functional motifs occurring throughout the target tissue. For those of you familiar with sparse coding, a sparse functional basis is simply a basis for a function space rather than a vector space, as is generally assumed in the field of sparse coding. Analogous to sparse coding, a sparse functional basis corresponds to a set of basis functions that define a family of functions in which each function in the family can be defined as a linear combination of the basis functions such that only a small number of the weights that define the linear combination are non-zero. In subsequent slides, we introduce additional concepts that will be useful in understanding the importance of this sparse functional basis. The concepts are found in linear systems theory, but some neuroscientists may be familiar with the ideas from writing programs in languages that support matrix vector operations from linear algebra, or their use in the widely used and unreasonably successful class of artificial neural networks known as convolutional neural networks. Since this presentation is intended for researchers from diverse backgrounds, and the model and method of learning it are sufficiently intertwined, we'll take a few minutes to review the basic terminology and technology relating to convolutions and convolutional neural network technology. To understand how we intend to learn and then apply a sparse functional basis in a mesoscale model, it will help if you are somewhat familiar with the terminology employed in working with linear filters, convolving a mask or kernel matrix with an image, and training convolutional neural networks. A simple grayscale image is generally implemented as a two-dimensional matrix. However, when an image is encoded as the input layer in an artificial neural network, it represents a vector in a vector space of dimension equal to the number of pixels in the image. When dealing with high-dimensional inputs such as image, it is impractical to connect all the units in the input layer to all of the units in, say, for example, a subsequent hidden layer. Instead, we connect each unit in the hidden layer to only a local region of the input layer. The spatial extent of this connectivity is a hyperparameter called the receptive field or filter size of the units in the hidden layer. Substitute the word neuron for unit, and the same terminology is applied to neural circuits, especially circuits organized as topographic maps, such as are found in the retinotopic maps appearing in the ventral visual stream of mammalian neocortex. The size of the hidden layer is determined by the receptive field and the spacing or stride between the local regions assigned to hidden units. In image processing, a kernel or convolution matrix is a small matrix that serves as a filter on the input image used for edge detection, image blurring, or other transformations. These transformations are accomplished by means of a convolution between a kernel and an image. A collection of such filters is called a filter bank. The term filter kernel or kernel function is generally employed when discussing the obvious generalization of convolution. In many image processing tasks, the kernel function is the dot product of the convolution matrix and a filter size region of the target data, corresponding to a matrix, volume, or some other form of structured data. In a convolutional neural network, the entries in the convolution matrix are free parameters that are learned via backpropagation. When computing the convolution of an image with a filter, we run a sliding window over the image, applying the kernel function to the kernel at each receptive field-sized local region of the input layer as defined by the filter size and stride parameters, resulting in a filtered image the same size as the hidden layer. The coordinates of the local regions constitute a grid spanning the input layer, a 2D grid in the case of a simple grayscale image, or 3D grid in the case of mesoscale volumetric data. Learning a sparse basis involves searching for an optimal combination of basis vectors for representing a vector space, or, in our case, a function space. The loss function is typically factored as a linear combination of the reconstruction error and a sparsity-inducing penalty function. The latter is generally based on various norms similar to those employed in compressive sensing. The L0 norm is just the number of non-zero elements in a vector, and hence is integer valued and not differentiable. And so optimization is often carried out by some variant of the analysis synthesis method used for solving convex optimization problems. The L1 norm, also known as least absolute differences, can be used with gradient methods and consist of minimizing the sum of the absolute differences between the target and the reconstruction of the target as a linear combination of the basis vectors. To get some perspective, consider this schematic rendering of the basic model. The graph enclosed in the circle labeled microcircuit represents the neural activity of the neural circuitry of the target organism, which, owing to the limitations of current state-of-the-art recording technology, might, at least in the near term, correspond to a fly, zebrafish, or some interesting subcircuit, such as the visual or olfactory system of a fly, or the entire or the striate cortex in mammals. The input can be thought of as the stimuli that the organism is exposed to in the process of the experimentalist carrying out some experimental protocol. The sensory environment for small organisms is likely to include chemosensory, thermosensory, and visual stimuli, as well as precisely targeted optogenetic stimulation of individual neurons and neuronal groups. The output corresponds to the observed behavior. Typically, behavioral data is acquired using high-speed cameras generating thousands of frames per second. The resulting video is then analyzed to generate a more compact, mathematically precise account of behavior, often by using Fourier analysis and spectrograph algorithms to reveal microscale behavioral modes that allow a more detailed account in keeping with the temporal resolution of the functional recordings. In addition to inputs and outputs that relate the macroscale behavior of the whole organism to its external environment, we are also interested in the microscale inputs and outputs of smaller neural circuits available from the functional data as measurements of calcium flux or local field potential. Recordings of neural activity at the cellular resolution enable us to evaluate proposals for decomposing large circuits comprised of many neurons into modular networks comprised of relatively few components, each one accounting for a subset of neurons in the larger circuit. This graphic illustrates the idea of a functional module that accounts for a subcircuit of the full microcircuit. Each such module has a domain that determines its inputs and outputs, an interface that accounts for its functional relationships with other modules, and an artificial neural network that defines its implementation. Each network corresponds to an instance of a class of configurable neural networks. We refer to these configurable neural networks as shallow, deep neural networks. They are deep in that they utilize the same basic component layers found in current generations of deep neural networks, and shallow in that they are comprised of a small number of component layers, each well known to neuroscientists because they were either discovered by neuroscientists studying the brain or inspired by the work of neuroscientists. Examples include divisive normalization, max pooling, and the sigmoidal, logistic, and rectified linear units often employed in hidden layers. The motivation for using shallow deep neural network architectures is that functional module networks are intended to serve as computational explanations of local neural function in our model. And given that we understand each of the component layers, it is relatively easy to explain what's going on in a shallow network in much the same way that that a short computer programs can be written in conventional programming languages are easy to understand if one steps through the program line by line, even if the program manifests complex behavior, as in the case of a short program implementing the one-dimensional logistic map that manifests chaotic behavior. While we have some way to go in mathematically understanding the unreasonable effectiveness of deep artificial neural networks, software engineers and machine learning researchers who routinely work with such networks can design, test, debug, and explain deep neural network architectures constructed from standard components, and, at least for shallow networks consisting of relatively small numbers of layers, can provide a reasonably intuitive characterization of their behavior. This is a schematic representation of the processing pipeline for Project Mindscope at the Allen Institute for Brain Science. Project Neuromancer at Google is working in collaboration with the Allen Institute on the parts of the Mindscope project emphasizing connectomic reconstruction that are shown as light gray boxes in the graphic. The two boxes highlighted with a red triangle represent the focus of the work described in this presentation. For you science fiction fans, Neuromancer is the title of a classic cyberpunk novel by William Gibson. I'm showing you this slide not because I'm going to explain it in any detail, but rather because I wish I could describe the class of mesoscale models and related machine learning technology for inferring these models as neatly and succinctly as this slide demonstrates. Alas, it's not as easy as it might seem, and I suspect that the next slide is the best I can hope for and still have any hope of providing a complete picture. To compensate, I will spend more time on this slide than any other in attempting to give you a comprehensive understanding of the model and related inference machinery. This slide describes the conceptual and technical interplay between the class of mesoscale models introduced in this presentation and the network architecture used to learn such models. The four elements in the middle sequence relate to the primary conceptual steps leading from the acquisition of training data to inferring mesoscale models. The top row lists some of the most important neurobiological desiderata involved in developing the class of mesoscale models considered here. The bottom row consists of some of the most important technological desiderata leading to the design of the network architecture for learning these models. I will focus on the middle row, advancing from left to right, and referring to elements in the top and bottom rows as required. The leftmost graphic in the middle row describes the data required for learning. The top half of this graphic represents the structural data. On the left is a sample electron micrograph of the neural tissue used to construct the connectome graph. The dense packing of neuropil is clearly evident in the micrograph. On the right is a reconstruction of the neuropil in the tissue, including the complete elaboration of the axonal and dendritic arbors. The structural data also includes cell types, morphology, and information about synapses such as vesicle counts that can be used to estimate the strength and valence, excitatory or inhibitory, of each synapse. The bottom half of this graphic represents the functional data, where here we assume that at a minimum we can record from every neuron in the target tissue. In the upper left-hand corner you see a juvenile zebrafish which, during the larval stage of its development, is essentially transparent. On the right are shown what are called rasters, corresponding to the recorded activity in each neuron. The terminology reflects the fact that whole brain functional imaging at cellular resolution using state-of-the-art light sheet microscopy requires scanning the 3D volume. The conventional raster representation represents in a loss of spatial context, which in the case of analyzing small tissue samples and in the absence of structural data is often not a limiting factor. Each full scan covers the entire volume and represents a single 3D slice in time, despite the fact that scanning takes time and so earlier points in the scan lag behind later ones. With parallel scanning at high frame rates, this lag could in principle be partially compensated for, but currently it is not, since neither parallel scanning nor high frame rates are technically feasible at this time. Locality is an important functional cue, and so instead of separate rasters for each neuron, we represent each complete scan of the tissue as a point cloud embedding 2-photon excitation fluorescent point sources having the same geometry as the static connectome embedding. Shared geometry facilitates the alignment of functional and structural data, allowing us to relate functional activity to the specific circuits that give rise to it. The sequence of all such structurally aligned point cloud representations constitutes a time series that serves as input to the network architecture for learning mesoscale models. The first graphic from the left in the topmost row depicts a small circuit with 3 neurons and 4 shared synapses. The 4 synapses and one of the 3 neurons are enclosed in a circle representing a cross-section of a spherical subvolume of the microcircuit connectome embedding space. The 4 synapses and their corresponding pre- and postsynaptic neurons constitute a subgraph of the full connectome. It is this subgraph that will serve as a starting point for defining the domain of a given functional module. The first graphic on the left, in the bottommost row, illustrates how this graph is represented in the learning architecture. The input layer for a mesoscale model corresponds to the neural state vector at time t. The output prediction layer represents the neural state vector at time t plus 1. Since we currently can't record from all of the synapses in any reasonably complicated tissue, synapses remain implicit, their weights corresponding to hidden variables that we attempt to learn from a combination of neural circuitry available in the connectome and functional recordings of their pre- and postsynaptic neurons. This structural and functional information could allow us to infer synaptic strength, given that we record from all of the neurons simultaneously. However, more likely than not, the problem is significantly under-constrained, and the best we can hope for is a rough estimate. The second graph from the left in the topmost row illustrates the transmission response protocol used to combine structural and functional data in order to capture the model dynamics in a series of transmission response graphs, each of which is a subgraph of the connectome graph, with all of the vertices in the connectome graph and a subset of the edges. The graphic shows a toy connectome graph. We assume that all and only those synapses present in the microcircuit are registered as edges in the connectome graph. We construct the sequence of transmission response graphs as follows. Divide the interval over which the functional data is collected into subintervals of some fixed duration. For each subinterval P, construct a transmission response graph G such that for each edge E in the connectome graph, E is an element of the set of edges of G, if and only if the functional data collected over the interval P supports the hypothesis that an action potential is propagated across the corresponding synapse from the pre to the postsynaptic neuron during the interval P. The second graph from the left in the middle, in the middle row, depicts a microcircuit connectome graph embedded in a 3D volume, and a spherical subvolume corresponding to a convolution receptive field. Applying the terminology of convolutional neural networks, a functional basis corresponds to a set of functional modules. The kernel for a functional basis filter corresponds to the configurable network associated with the module. The free parameters of the kernel correspond to the configuration parameters that determine a fully instantiated configurable neural network. The kernel function applies this network to functional domains extracted from the local regions. The input layer of a mesoscale model is a 3D embedding of a transmission response graph, not a simple matrix or higher dimension tensor. The grid is defined by 3D coordinates spanning the volume corresponding to the neural microcircuit. The local regions corresponding to receptive fields are spherical subvolumes of a fixed diameter separated with a stride substantially less than the diameter. The sliding window operator is implemented by a highly parallel distributed spatial index based on KD trees, as illustrated in the second graphic from the left on the bottommost row. Training a mesoscale model involves learning a set of configuration parameters for each functional module network, just as we learn the convolution matrix for each filter in a traditional filter bank. We also learn the sparse weights that define the linear combination of network outputs at each location in the input graph embedding, and finally, we learn a separate functional module interface for each combination of module and receptive field in the 3D embedding of the transmission response graph. We don't expect the fixed diameter spherical subvolumes defined by the fixed 3D grid of coordinates to exactly partition the volume into functional domains. The filter diameter and stride that determines the grid of points are supplied by the researcher constructing the model and only serve as a proposal or upper bound on the extent of functional domains, and by so doing constrain search. Each spherical subvolume encloses a unique maximal subgraph with a variable number of vertices and edges. Since adjacent spherical subvolumes overlap, their enclosed subgraphs may share some vertices and edges. We think it very likely that in some cases, multiple functional modules will cover different portions, that is, subgraphs or sub-subgraphs, of the subgraph enclosed by any given spherical subvolume. This complication highlights another issue relating to variability in the size and constitution of these maximally enclosed subgraphs. The parameters of each configurable network instantiating a domain module are set globally. Each network is expected to represent a distinct functional motif, and some of these networks will contribute to predicting portions of the neural state vector in different locations throughout the target microcircuit. But the enclosed subgraphs will vary, and different regions of a microcircuit are likely to manifest different spiking frequencies, signal attenuation, and noise characteristics. The solution is to learn a local interface for each functional module or enclosed subgraph pair. The interface for a given module and enclosed subgraph aligns the inputs and outputs by selecting some neurons for inclusion in the module domain and rejecting others, modulates and shapes the local signal characteristics, and matches impedance between the specified module and the selected inputs and outputs. This is analogous to matching one amplifier with different speakers and input devices. The third graphic from the left in the middle row represents an inverted architecture for a convolutional neural network, with the input on the top and the output on the bottom. The cube on the top right is the static connectome graph embedding essentially a topographical map of the neural tissue. The sequence of cubes on the top left represents the time series of point clouds embedding the two photon excitation fluorescent point sources with the same geometry as the static connectome embedding. The penultimate layer fuses the static structural data and the dynamic functional data to generate the series of transmission response graph embeddings described earlier. The remaining layers ingest each transmission response graph in sequence, updating the neural state vectors and integrating new data to estimate the error in predicting the next neural state vector. The layers following the functional structural fusion layer are responsible for adjusting the global functional module configuration parameters as well as the parameters that define the local functional module interface layers and assign point sources to each functional module domain. These operations are not performed sequentially as shown in the diagram, but rather all these adjustments are carried out simultaneously depending on end-to-end stochastic gradient descent to optimize all of the model parameters in parallel in searching for an optimal solution. The third graphic from the left in the topmost row shows a conventional abstract neural circuit model. We argued earlier that models such as these provide useful insight concerning the role of different cell types and connection patterns in relatively small circuits, but fall short in terms of reaching much beyond the level of individual neurons, or explaining the aggregate behavior of large neuronal ensembles. The third graphic from the left on the bottom row represents an instance of the class of configurable neural networks proposed here, showing the global parameters that specify functional modules comprising a sparse functional basis and allowing us to identify the functional motifs, and the local parameters that specify functional interfaces allowing us to adapt those functional motifs to different subcircuits. In addition to providing a computational predictive model accounting for the neural activity of relatively complex neural mechanisms, the model consists of modular, reusable components, each of which represents the aggregate behavior of some functionally separate neural ensemble, and supports a wide range of in silico experiments designed to test the stability and generality of particular subclasses of models. The graphic on the far right in the middle row illustrates the layers of the convolutional mesoscale model architecture that are responsible for learning a sparse functional basis. We've touched upon most of the technical features of this architecture. However, those features are among the most important contributions of this work, so it's worthwhile highlighting the main points with this graphic as a visual aid. We'll postpone mention of the graphics on the far right of the first and third rows, since they'll be a little easier to explain once we look at the learning architecture. Making all three of these a little larger and more detailed will likely help us out as well. The network shown here consists of 7 layers and represents only a part of the neural network architecture for learning a mesoscale model. The 2 layers labeled Roman numeral 2 and 5 and shaded green encode the complete neural state vector at time t and t+1 respectively. Neural state vectors are derived from the functional data encoded in the sequence of transmission response graphs that combine both structural and functional data. The bottom layer, shaded in purple, encodes a set of spatially and temporally localized features computed from the current transmission response graph that have proved to be effective in identifying functional motifs. These features assist in learning the functional domain boundaries. The layer labeled Roman numeral 3 and shaded orange is divided into sublayers, shown here by dashed lines, each sublayer encoding the local parameters for one functional basis filter. Layer 4 is a convolutional layer, shown here with 3 functional basis filters, each one associated with a unique functional module and its corresponding parametrically configured neural network. The units in the penultimate layer, labeled Roman numeral 5, constitute the output layer of the model, divided into sublayers shown separated by dashed lines, 1 layer for each basis filter. The final layer scores the accuracy of each basis filter at each location. The basis filters are responsible for reconstructing the neural state vector of each recorded neuron. If there are 17 filters, each neuron is modeled by a sparsely weighted linear combination of 17 filters, so that only a few filters contribute significantly in accounting for any given neuron. Once trained, each filter applies the same globally configured network everywhere, but employs a locally configured interface to adapt the globally defined module to local conditions. Each fully trained network represents a distinct functional motif. The system as a whole can be deployed as a powerful motif finder. The framework allows us to explore a number of interesting hypotheses. By experimenting with the sparsity-inducing penalty term in the loss function, it should be possible to explore questions concerning whether all cortical volumes compute the same function, or measure the diversity and distribution of function if it appears there is no single function. We postponed mention of two important concepts that were included as a graphical placeholder in slide 10 in order to take a closer look at the convolutional architecture for learning mesoscale models. The first of these concepts is depicted in this expanded graphic, illustrating how the maximally enclosed subgraphs associated with a receptive field domain proposals are apportioned to multiple functional modules. The graphic illustrates the functional basis filters, how the functional basis filters decompose the connectome graph and associated point sources into functional domains. Each of the large dashed and solid circles represent a spherical subvolume of the connectome embedding space. When training is complete, each subvolume and each point source will be claimed by exactly one basis filter. In the graphic, 3 color-coded filters are shown, claiming a total of 6 of the 14 subvolumes. The stride of the sliding window convolutional operator is half the diameter of the receptive field subvolume. Note that with the exception of B, none of the filters include, and therefore are responsible for modeling, all the point sources in their subvolume. The second of the two concepts, postponed from slide 2, is depicted in this expanded graphic, illustrating how functional modules are evaluated and assigned the responsibility for predicting different parts of the neural state vector. 3 functional module networks are shown in green along with their respective interface layers. The top 2 layers of each stack of layers, shown in green, compute signals measuring the predictive accuracy of each functional module. During backpropagation, these signals are fed back and combined with additional local network features to assign cells to functional domains. These features are derived from the local properties of the static connectome graph and summary statistics of the functional time series characterizing the mutual information of adjacent subcircuits. The components shown in blue correspond to multi-layer networks that learn how to apportion cells to functional domains. These layers adjust location-specific parameters in the configuration layer and use the updated information to restrict functional module domains accordingly. The inset graphic underscores the fact that functional domain assignments often involve restrictions with the red dashed lines depicting cells that are not included in the domain. The material covered in this presentation is demanding in large part because it spans several disciplines, including machine learning, computer science, molecular and cell biology, computational neuroscience, and software engineering. I tried my best to make the material as clear as possible by avoiding jargon, defining my terms carefully, and using easily accessible graphical representations to convey concepts that might otherwise require a mathematical treatment in speaking to a specialized audience. This is an almost comically busy slide, hence the tongue-in-cheek title. It was originally intended as exactly that, comic relief in an otherwise technically challenging presentation. However, at one of my lectures, a student mentioned that this graphic could serve as the index for a pictographic dictionary, with each of the small graphical icons linked to a term in the glossary or expanded explanation in a longer exposition. And so I created just such a document and provided a link to it on the first slide in this presentation. Some of the icons link to captions in the current draft of a white paper I'm preparing for publication. The draft is still in flux, and I don't guarantee that all of the prose is entirely coherent at this stage, but you may find it interesting as a snapshot of my thinking. On the course website, I've included the slides used in this presentation, as well as a set of supplementary slides providing additional technical detail that you can access by clicking on the slide heading. If you are challenged for a username and password, enter the username brain and password maker to access the material. This presentation is intended as an overview of one possible approach to building mesoscale models. As pointed out in the introductory slides, the work described here spans more than just a class of models. It is a research plan for conducting large-scale, data-driven neuroscience that includes technology, tools, models, and a methodology for moving forward on some of the most difficult problems facing neuroscience today. It is not the last word by any means, and I sincerely hope it will widen the discussion to include new ways of thinking about these problems and invite an even broader range of scientists, engineers, and technologists to help in solving them. I'll leave you with just a few of the ideas we're thinking about for how to extend this network.